No problem.
Correct.
Sure. Okay, so I'll try and finish by like Isha time. That's okay. So my name is Areez Shafqat. This is a lecture on the feed fast cycle. Um, so let's begin this lecture by basically talking about why we eat. That's what this diagram is basically showing us. So we eat because food has macronutrients in the form of carbohydrates, proteins, and fat. Carbohydrates get broken down into glucose. Refer to your GI lectures for that. Then glucose gets broken down into pyruvate by a process called glycolysis. Pyruvate goes into acetyl-CoA by an enzyme called PDH, right? Proteins. Okay, so why we eat is because food has macronutrients in the form of carbohydrates, proteins, and fat. Carbohydrates get broken down into glucose and pyruvate by a process called glycolysis. Pyruvate then gets converted into acetyl-CoA by an enzyme called PDH, right? Proteins as well, the same thing, they get broken down into amino acid and eventually acetyl-CoA. Fats, the same thing. Fats are ingested in the form of triglycerides. They get broken down into fatty acids. Fatty acids get broken down into acetyl-CoA by a process called beta oxidation, okay? So as you can see, all of the food that you eat, the macronutrients that you ingest, all of them go funnel into acetyl-CoA, which is used in the electron transport chain in the TCA cycle to generate ATP. So this is why you eat, okay? Now, 50% of the energy in the electron transport chain is used to make ATP, the other maintains body temperature. How we will transition into discussing regulation is by this curve here. This is the glucose tolerance curve. It shows you time on the x-axis, blood glucose on the y-axis. At minute 0, the guy ingests a carbohydrate meal. Blood glucose goes up and then it comes back down. So at this side of the glucose tolerance curve, at this part, what hormone is activated? Because blood glucose is high, that stimulates pancreatic beta cells to secrete insulin. Insulin decreases blood glucose. If you know that much biochemistry, you can understand that insulin, because it decreases blood glucose, will stimulate pathways which use up glucose. Either insulin will stimulate pathways which use up glucose in energy, either insulin will stimulate pathways which store glucose as glycogen or as fatty acid. Then you can understand also that in diabetes, you don't have the effect of insulin. In type 1 diabetes, you don't have enough insulin. In type 2 diabetes, you have the insulin, but the cells don't respond to it properly. So in type 2 diabetes, you won't have those effects of insulin, glucose lowering effects of insulin. And so in diabetes, you would have hyperglycemia, okay? However, if you see in diabetes, glucose does come down eventually. And that's because in your body, there are insulin-independent cells which don't require insulin to take up glucose. The two cells which you need to know, the two insulin-independent cells that you need to know are the brain and the red blood cell, okay? So that's all you need to know from this slide. Why you eat is because macronutrients funnel down into acetyl-CoA, which is used to make ATP. Then in the glucose tolerance curve, you can see that insulin activates metabolic pathways, which use up glucose. In diabetes, you don't have this, so you have hypoglycemia, hyperglycemia. But in diabetes, glucose does eventually come down, and that's because of insulin-independent cells. The two most important insulin-independent cells you have to know are the brain and the red blood cell, okay? Now, the doctor which gave you this PowerPoint put this down. I'm going to sort of summarize all of this into understanding regulation. A lot of people don't really understand regulation, which enzymes is insulin activating, which enzyme is glucagon activating. To understand that part, all you have to do first is to ask what condition is the body in, okay? Is the body well fed or is the body fasting? If the body is well fed, that means blood glucose is high. If the blood glucose is high, what hormone will be active? If the blood glucose is high, insulin will be active. So, because insulin will store glucose or it will use glucose. Okay? How are these pathways regulated? How does insulin exert its effect? It exerts its effect by gene expression or by dephosphorylation. The dephosphorylation effect is quicker. The gene expression effect is slower. Okay? So how to understand regulation again? Let me discuss the alternative scenario. Let's say the body is in a starving condition. So what hormone will be active? The hormones which will be active will be glucagon, will be cortisol, which will be epinephrine. These pathways, because in a fasting state, glucose is going down, these pathways will increase blood glucose by producing glucose. So glucagon, cortisol, epinephrine will activate pathways which produce glucose. pathways like gluconeogenesis, pathways like glycogenolysis, breakdown of glycogen, fatty acid breakdown, and then beta oxidation as well, and ketogenesis. Glucagon, like insulin, also acts at gene expression. This effect is slower, and it acts at the phosphorylation, dephosphorylation level. Glucagon specifically causes phosphorylation. Insulin causes dephosphorylation. Okay. So let me just give you a few examples. This is the pathway of glycolysis. Okay. We'll apply the same questions to the glycolysis pathway. So what condition must the body be in for glycolysis to happen? Glycolysis is a pathway which uses up glucose. That means to use up glucose, your body needs to have a lot of glucose. That means glycolysis will occur in a well-fed state. In that state, what hormone is active? Yeah, exactly. So in that state, what hormone is active? Insulin. That means insulin will activate glycolysis. Then how does insulin activate glycolysis? Insulin activates this enzyme glucokinase. Insulin also activates phosphofructokinase. Insulin activates glucokinase at the gene level. This effect is slower. Insulin activates PFK2 by dephosphorylation. This effect is immediate. Okay. You don't need to know this pathway. This was MOL1, but this is just to give you an example on how to understand regulation. It's the same with glycogen synthesis. Glucose is here. It gets converted into glucose 1-phosphate and then made into glycogen. For glycogen storage to happen, what condition must your body be in? In order to store glycogen, you must have a lot of glucose. That means to make glycogen, you should be in a well-fed state again. Yes. That means what hormone should be active? Insulin. Insulin activates an enzyme called glycogen synthase by dephosphorylation, then to produce glycogen. The alternative pathway is glycogen breakdown. What condition must your body be in for glycogen to be broken down? Starving. That means glucagon will be active. Glucagon activates an enzyme called glycogen phosphorylase by phosphorylation, which causes glycogen breakdown. So by asking these 4 questions, you can understand regulation quite well. Okay. This pathway you have in MOL2, so I'll go into a bit more detail here. Okay. So glucose enters the bloodstream. Let's say you've eaten a high carbohydrate meal, you're well fed, glucose is high in the blood, glucose enters the liver. Okay. Glucose gets converted into pyruvate by glycolysis. Pyruvate gets converted into citrate during the Krebs cycle. As the liver is sort of utilizing glucose in this way, citrate is accumulating. The accumulated citrate will go out into the cytoplasm and generate acetyl-CoA. Acetyl-CoA gets converted to malonyl-CoA, and then malonyl-CoA get converted into fatty acid. The fatty acids are joined to glycerol to form triglycerides. Okay, so if I ask you what hormone would activate this pathway, you should ask these 4 questions again. Fatty acid synthesis would occur in a well-fed state. That means the body would be releasing insulin. That means insulin would activate fatty acid synthesis. What enzymes does insulin activate? Insulin activates acetyl-CoA carboxylase at the gene level. Insulin activates fatty acid synthase as well. Okay, so this is fatty acid synthesis. Let me ask you guys a question. There are 2 storage forms of energy in your body. One is glycogen, one is fat. What is the major storage form of energy in your body? Is it glycogen or is it fat? It's fats. Do you know why? Because fats are hydrophobic. So glycogen is osmotic because glycogen contains oxygen bonds and carbon bonds. That means it's polar. That means it holds on to water. So there's only a limited amount of glycogen you can store. Otherwise, the liver would swell up. And that's what happens in glycogen storage diseases, right? Glycogen accumulates and the liver swells up with hepatomegaly. Fats are hydrophobic, so you can store as much of them as you like. So what your body does when you eat a high carbohydrate meal is that insulin activates glycolysis. Insulin also activates glycogen synthesis like we discussed. However, your body can only synthesize a limited amount of glycogen. Once that limited amount is reached, insulin activates fatty acid synthesis through this pathway because you don't want a lot of glycogen accumulating. Okay, so just to recap, you can only store a limited amount of glycogen in your body because it's osmotic. Fats, you can store them as much as you like. So therefore, when glycogen stores are full, insulin activates fatty acid synthesis by activating acetyl-CoA carboxylase and fatty acid synthase. Okay. This is a summary on lipoproteins. When you eat a meal, your meal contains fats. Those fats are broken down in the gut by pancreas and bile, by the pancreatic lipase. Those fats get absorbed into the enterocyte. They get remade and packaged in things called chylomicrons, okay? So fats, as they enter the enterocytes, they're packaged into chylomicrons. Chylomicrons contain a protein called B48. This protein is important for chylomicrons to go out into the lymphatics. So fats are absorbed into the lymphatics within chylomicrons. And then chylomicrons sort of move through the lymphatics, end up in the thoracic duct, then go into the bloodstream. In the bloodstream, chylomicrons kind of bump into HDL, okay? And HDL gives them ApoE and ApoC2. I've listed the functions of apoproteins here. ApoA is found in HDL. It activates LCAT. We'll look into that. ApoB, there's two types. B48 is found in chylomicrons. It's required for the exocytosis of chylomicrons into the lymphatics. B100 is found in VLDLs, LDLs, and IDLs. ApoC2 activates LPL, lipoprotein lipase. ApoE causes uptake of lipoproteins by the liver. You have to remember these functions. These are very important, okay? So let's get back here. Chylomicrons are in the bloodstream. They're bumping into HDL. HDL gives them ApoE and ApoC2. ApoC2 activates lipoprotein lipase, which extracts fatty acid and deposits it into the adipose tissue and the muscle, okay? So why is this important? Why am I discussing this? Because insulin activates lipoprotein lipase as well at the gene level. And this goes in line with the questions we were asking, right? Because this would be after a meal. So this is well-fed state. So insulin would be active. And active insulin would stimulate lipoprotein lipase at the gene level. The lipoprotein lipase would be expressed in the adipose tissue and in the muscle, the capillaries, and it would extract fatty acids from these chylomicrons. And the chylomicrons would start shrinking. Now those chylomicrons are called chylomicron remnants. They give back ApoC2 to the HDL. So now they only have ApoE and ApoB48. The chylomicron remnants then get taken up by the liver by ApoE, okay? At the same time, the last slide we looked at, this one, the liver is synthesizing fatty acid, and the liver is packaging these fatty acids and triglycerides within things called VLDLs, like this pathway here. Those triglycerides that the liver makes, it packages them in VLDLs. So now the liver starts releasing VLDLs, and they contain B100. The same thing happens with VLDLs. They bump into HDL. HDL gives them ApoC2 and ApoE. ApoC2 activates lipoprotein lipase, which is activated by insulin. Then lipoprotein lipase extracts fatty acid. The VLDL starts shrinking. Now it's called an IDL. Now it doesn't need the ApoC2 anymore, so it gives it back to HDL. Now it only has ApoE and ApoB100. Now it has two options. Either it can get taken up by the liver through ApoE, or it can give back ApoE to HDL. Now, IDL with B100 only is called LDL. Okay, so LDL only has ApoB100. The LDL has two options now. LDL can get taken up by the liver, by the LDL receptor for B100, or LDL can deposit in the periphery because peripheral tissues express the LDL receptor. And that's why LDL is considered bad cholesterol, okay? Because it deposits cholesterol in the periphery, because your peripheral tissues have LDL receptors. Another reason why LDL is bad is because it only has B100, and this can get oxidized. Once it gets oxidized, this protein is recognized by macrophages, which take it up through their scavenger receptor and become foam cells. And this plays a role in atherosclerosis, okay? Now LDL, because of its B100 and LDL receptor, it deposits cholesterol into peripheral tissues. HDL, what it does is that it picks up this cholesterol in peripheral tissues and through an enzyme called LCAT, deposits it in the form of cholesterol esters within HDL, and then takes it back to the liver, okay? So that's why HDL is considered good cholesterol. It picks up cholesterol from the periphery because of an enzyme called LCAT, which is activated by HDL, which is activated by ApoA. And this allows HDL to take cholesterol back to the liver. So LDL takes cholesterol to the periphery, HDL brings it back to the liver. Okay. I'm going through these pathways because we'll sort of tie them up together at the end, and I want that to make sense, okay? This is gluconeogenesis. You took it in MOL 1, so I won't go into details, but what condition should the body be in for you to do gluconeogenesis? Gluconeogenesis produces glucose, right? So you should be in a starving condition. In a starving condition, the body is releasing glucagon and cortisol. So glucagon and cortisol activate gluconeogenesis by activating PEP carboxykinase, this enzyme at the gene level. You don't need to know that for now. All I want you to know from gluconeogenesis, and this is important, is that gluconeogenesis requires pyruvate, and that goes back into glucose. Okay, so you need a source of pyruvate. That's the first thing. The second thing is that gluconeogenesis requires ATP. Okay, it requires this, it requires this step here. So there must be a way of generating ATP to fuel gluconeogenesis. Okay, so the two things I need you to know from gluconeogenesis: number one, you require a source of pyruvate. Number two, you require ATP. You require a source of ATP. This is important for you, this pathway, because this is in mode 2 as well. Now, this is triglyceride breakdown, fatty acid breakdown, okay? Lipolysis. What condition must the body be in for you to break down adipose tissue? You must be starving, right? So which hormones should be active? Glucagon, cortisol, and epinephrine. Epinephrine and cortisol activate an enzyme called hormone-sensitive lipase. This enzyme breaks down triglycerides into glycerol and fatty acid. The glycerol goes back into the liver, and glycerol is an intermediary of glycolysis. Look here, this is glycerol here. So glycerol from the adipose tissue goes back to the liver, and glycerol is made back into glucose. Okay, so this glycerol here is a source of glucose. It's a glucogenic substrate. What happens to the fatty acid? The fatty acids go into the bloodstream. They're bound to albumin. Fatty acids go into the liver. They can get oxidized to form acetyl-CoA and then the citric acid cycle, and then they can be made to form ATP. Okay, so now why is this important? Because I told you in this slide that gluconeogenesis requires ATP. Where do you get the ATP from? You get the ATP from beta oxidation of fatty acids. Okay, this pathway here, this whole pathway during the starving state, fatty acids are triglycerides are broken down, fatty acids go into the liver, get burned in the Krebs cycle to make ATP. This ATP fuels gluconeogenesis. Okay, with prolonged fasting though, acetyl-CoA will be accumulated because now you're breaking down a lot of fat. So acetyl-CoA will accumulate and a lot of it will get shunted into ketone body production. Why is this important? Fatty acids are bound to albumin. That means fatty acids can get taken up by the liver for energy. Fatty acids can get taken up by muscles for energy, but fatty acids cannot cross the blood-brain barrier. Because fatty acids cannot cross the blood-brain barrier, the brain cannot utilize fatty acids during fasting. Okay, that's why ketone bodies are important during fasting, because by producing ketone bodies, the brain begins to utilize ketone bodies with fasting. So by producing ketone bodies, you provide an extra fuel for the brain. Okay, I hope this makes sense. This is very important. It does come in exams, by the way. So just to recap, triglycerides, to break down triglycerides, you need to be in a fasting condition. That means glucagon is active. That means epinephrine is active. That means cortisol is active. They activate hormone-sensitive lipase by phosphorylation at the gene level. Breakdown of triglycerides produces glycerol. Glycerol goes back to the liver, where it's made into glucose by gluconeogenesis. Fatty acids go to the liver to produce acetyl-CoA and ATP. This ATP is used to fuel gluconeogenesis. Excess acetyl-CoA is produced into ketone bodies, which can be used by the brain, and that's important. Okay. Now, any questions so far? Can we pause the recording for a second? That's okay. Just to recap, what are the two pathways by which insulin and glucagon regulate enzymes? They can either regulate enzymes at the gene level. This is a slow response. They can regulate enzymes by phosphorylation, dephosphorylation. This is an immediate response. Insulin activates enzymes by dephosphorylation, glucagon by phosphorylation. Okay. Insulin activates enzymes insulin activates pathways which use up glucose. These pathways include glycolysis by glucokinase and PFK2, glycogenesis by glycogen synthase, fatty acid synthesis through acetyl-CoA carboxylase and fatty acid synthase, and fatty acid deposition or adipose fat deposition by regulating the enzyme lipoprotein lipase. Glucagon, cortisol, and epinephrine activate pathways which produce glucose or ketones. So they upregulate glycogenolysis or glycogen breakdown, gluconeogenesis, lipolysis or fatty acid breakdown, and ketogenesis. Okay. How would this balance be disturbed in diabetes? That's an important question, because in diabetes, you don't have insulin. That means the action of these hormones is amplified because the balance between insulin and these hormones, there's a balance. In diabetes, this balance shifts in favor of glucagon, cortisol, and epinephrine. So what would happen? You would have hyperglycemia, you would have lipolysis and elevated fatty acids, and you would have ketone genesis. So all of the metabolic abnormalities you see in diabetes can be explained with some biochemistry here, okay? Because of a lack of insulin effect. Okay, so now let's sort of connect all these pathways together, okay? So let's say you eat a high carbohydrate meal. Now let's start with insulin independent tissues. The ones I told you to remember were the red blood cell and the brain. You have to know that the red blood cell and the brain express GLUT1 and GLUT3, okay? These transporters are not affected by insulin. Insulin has no effect on them. That means regardless of whether you're in a fasting condition, regardless of whether you're in a welfare state, glucose is entering the red blood cell and the brain at a very steady pace because these GLUT transporters are insulin independent, okay? Let's move on. Let's talk about the liver. You've eaten a high carbohydrate meal. Glucose starts entering the liver through a transporter called GLUT2. It's important to know that GLUT2 is not insulin dependent. The insulin doesn't tell the liver to take up glucose. The insulin is telling the liver what to do with the glucose. The insulin isn't telling the liver to take up glucose. So GLUT2 is an insulin independent transporter. In exams, they will ask you whether GLUT1 or GLUT2 or GLUT3 are insulin dependent. They're not insulin dependent, okay? These transporters are insulin independent. To activate glycolysis here. So this is glycolysis, glucose to pyruvate. Then pyruvate generates acetyl-CoA, and that generates ATP. Once the ATP is enough, insulin tells the liver to synthesize glycogen by activating the enzyme glycogen synthase by dephosphorylation. Once the glycogen stores are full, because you can only store a limited amount of glycogen, right? Because glycogen is osmotic. All of that extra glucose gets made into fatty acids because insulin activates fatty acid synthase and insulin activates acetyl-CoA carboxylase. These fatty acids are joined to glycerol to form triglycerides. They're again packaged in VLDLs and they're sent out into the bloodstream. Okay. Now, insulin now it basically told the liver to generate VLDLs. At the same time, insulin goes to the adipocyte and tells the adipocyte, hey, I've told the liver to produce fatty acids. I want you to upregulate LPL, lipoprotein lipase, to allow, and then when the adipose tissue upregulates this enzyme allows it to extract fatty acids from VLDLs and chylomicrons in the form of fatty acids. The glycerol goes back to the liver. And now, it's important this concept is quite important actually. Insulin upregulates LPL on adipose tissue, causes uptake of fatty acids. It doesn't tell the adipocyte to also take up glycerol because glycerol is going back to the liver. So where does the adipocyte get the glycerol from? Insulin upregulates GLUT4, okay, on the adipocyte. Insulin upregulates GLUT4, and then a lot of glucose starts entering the adipocyte. When this glucose enters the adipocyte, it gets made into pyruvate by glycolysis. A byproduct of glycolysis is glycerol 3-phosphate, right? And so this glycerol is used to make triglycerides. So again, let me explain again. At the adipocyte is taking up fatty acids by LPL, but it's not taking up glycerol. So what insulin does is it upregulates GLUT4, okay? And this causes a lot of glucose to start entering the adipocyte. This glucose is made into pyruvate by glycolysis. A byproduct of glycolysis is glycerol. And so you're using this glycerol to join fatty acids and make triglycerides, okay? So insulin is kind of doing both things, is doing everything basically. Insulin is also having effects on the muscle. Insulin is upregulating GLUT4 on the muscle, stimulating glucose uptake into the muscle. Insulin is stimulating glycogen synthase in the muscle to cause glycogen production. Insulin is also stimulating amino acid production in the muscle. So insulin stimulates muscle hypertrophy, basically, muscle buildup, all right? Because insulin stimulates protein production in the muscle as well. So all of these pathways are what are happening when you're in a well-fed state. Insulin is going to the liver, telling the liver what to do with the glucose. Insulin is telling the adipocyte to upregulate GLUT4, to upregulate LPL. It's also telling the muscle to upregulate GLUT4, upregulate glycogen synthesis, to upregulate protein synthesis. Importantly, GLUT1, GLUT2, and GLUT3, they have no relationship to insulin, okay? You have to remember that. Now, in diabetes, you have none of this. So in diabetes, you don't have glycolysis, and the liver doesn't really know what to do with the glucose anymore because there is no insulin effect. You don't have LPL, you don't have GLUT4 expression. And so what happens? Hyperglycemia, because insulin is not being taken up by the adipocyte and the skeletal muscles. So you have hyperglycemia. Okay. Now, this is what the doctor said. Immediately after a meal, blood glucose rises and stimulates the release of insulin. There are three major organs for insulin, target organs for insulin: the liver, the muscle, and the adipose tissue. Insulin stimulates glycogen synthesis in the liver and the muscle. After these glycogen stores are full, insulin converts the excess glucose to fatty acids, and then that's made into triglycerides, because fat is the major storage form of energy in the body. Insulin then stimulates triglyceride synthesis in adipose tissue and protein synthesis in skeletal muscle, as well as glucose entry into both tissues. This is mediated by GLUT4, okay? Now, importantly, you need to know that the two tissues, the brain and the red blood cell, are insulin independent. The brain derives energy from glucose in both the well-fed and fasting state. Only in prolonged fasting does the brain start utilizing ketone bodies, because as I told you, the brain cannot utilize fatty acids because they don't cross the blood-brain barrier. However, you also have to note that the red blood cells, they don't have mitochondria. So the red blood cells use glucose anaerobically, i.e. glycolysis, for all their energy needs, regardless of whether you're well-fed or fasting, okay? So if you think about it, during a prolonged fast, when the brain switches to use ketone bodies, it's actually saving some of the glucose for the red blood cell, because the red blood cell can only utilize glucose. Now, this is a section from first aid, just to summarize for you the effects of insulin. Insulin is activating glucose transport in skeletal muscle and adipose tissue by GLUT4. Insulin is stimulating glycogen synthesis and storage by activating glycogen synthesis by dephosphorylation. Insulin stimulates triglyceride synthesis by activating fatty acid synthase and acetyl-CoA carboxylase. Insulin is also stimulating sodium retention in the kidneys. This is not important for you to know. Insulin stimulating uptake of potassium is not important for you to know. Now, these transporters are essential for you to know: GLUT1 in the red blood cells in the brain, GLUT2 in the liver, okay, and in the pancreatic beta cells, GLUT3 in the brain, also in the red blood cell. Brain utilizes glucose for metabolism normally and ketone bodies during starvation. The red blood cells, however, always utilize glucose because they lack mitochondria for aerobic metabolism. Okay, so now any questions about the well-fed state, or is all of this clear? Is it okay if you repeat GLUT2? Okay, yeah, sure. Now, it's okay with you. Okay, so it's easy to think that because insulin is acting on the liver, that is stimulating. Okay, sure. It's easy to think that because insulin is acting on the liver, that insulin is, okay, yeah. So insulin does not stimulate glucose uptake into the liver. Insulin tells the liver what to do with the glucose. Insulin tells the liver to carry out glycolysis. Insulin tells the liver to carry out fatty acid synthesis. Insulin tells the liver to carry out glycogen synthesis. Insulin does not tell the liver to take up glucose. Okay, so GLUT2 is insulin independent. So they might trick you in the exam and say that, you know, the insulin has so many effects on the liver, but glucose uptake isn't one of those effects. Okay, so GLUT2 is insulin independent. Does that make sense? Okay. So this is a question based on those GLUT transporters. So researchers are conducting investigating the relationship between glucose transport proteins and insulin concentration. So what they did here basically is they took different cells and they measured glucose transporter activity. And what they did was they increased insulin concentration constantly, okay? So they continuously increase insulin concentration here, and they measure whether the increasing insulin actually stimulates glucose uptake. So in this cell, glucose uptake, regardless of insulin concentration, is the same. However, if you look in this cell, as insulin concentration is increasing, glucose uptake is increasing. So what cell type is this? This would likely be the skeletal muscle or the adipose tissue. This would likely be the red blood cell or the brain, because this is likely GLUT1 and GLUT3, because insulin does not have an effect, right, on the glucose transporter activity. However, if you look at this one, insulin is stimulating glucose uptake. So this must be GLUT4. So this must be skeletal muscle and adipose tissue. And this is a question from step 1. So this, I'm just to show you that this actually does come in exams. Okay, so now let's move to the other side. Let's discuss fasting state, okay? So in fasting state, you have a decrease in blood glucose that stimulates glucagon production. Can liver be the answer to it? Yes, the liver can also be the answer. Yes, it can be. Every cell type in the body can be the answer. The kidney can be the answer, for example, because the kidney, glucose uptake in the kidney is not regulated by insulin, okay? So they might give you any cell, any organ or any tissue, and most of tissues are insulin independent, okay? But if it can't be the skeletal muscle and the adipose tissue. Okay, so now this is the fasting state, a decrease in glucose. Okay, a decrease in glucose stimulates glucagon release. Glucagon acts on the liver. Glucagon firstly acts on the liver by stimulating glycogen phosphorylase by phosphorylation, and that causes glycogen breakdown. Okay, glucagon also activates gluconeogenesis at the gene level. This is a slow response, and this is the gluconeogenesis pathway here. So now remember what I told you about gluconeogenesis, right? You need a source of pyruvate. So where do you get the pyruvate from? The first source of pyruvate is the red blood cell, because the red blood cell only carries out glycolysis. And so the end result of anaerobic glycolysis is lactic acid. This lactic acid goes to the liver and it gets remade into pyruvate. And this gets used for gluconeogenesis. Okay. The second source is the muscle, because in fasting conditions, you have muscle protein breakdown. All of those proteins and amino acids eventually get funneled into alanine. Alanine goes into the liver. It gives off its amino group to alpha ketoglutarate, and that goes into the urea cycle. The remaining carbon skeleton of alanine is pyruvate. Okay, so alanine comes from the muscle, from muscle breakdown. It gives off its NH2 group, and what's left is pyruvate. This is also used for gluconeogenesis. The third source is glycerol here, because as I told you, during fatty acid fat breakdown or via hormone-sensitive lipase, the glycerol goes to the liver and it gets made into glycerol 3-phosphate. This glycerol is used to make glucose, okay? So there are 3 sources of pyruvate for gluconeogenesis during starvation. The first one, lactic acid from the red blood cell. Number 2, alanine from the muscle. Number 3, glycerol from the adipose tissue. So that's the first thing you need to know. The next thing you need to know is what happens to the fatty acids. The fatty acids are released by hormone-sensitive lipase. This is activated by cortisol and epinephrine. The fatty acids go into the bloodstream where they're bound to albumin. They can't cross the blood-brain barrier. So you see there's no fatty acid uptake into the brain here. However, fatty acids can go into the muscles to make ATP by beta oxidation. They can go into the liver to make ATP by beta oxidation. This ATP fuels gluconeogenesis, right? However, during prolonged fasting, so much fatty acids are being broken down that acetyl-CoA starts accumulating. And so you shunt the excess acetyl-CoA into ketone body production. These ketone bodies can be used by the muscle for ATP, but importantly, they can also be used by the brain. When ketone bodies are used by the brain, they form acetyl-CoA, because as I told you, right, with the first slide where I was showing you why you eat, all of the macronutrients get funneled into acetyl-CoA for energy. So similarly, ketone bodies get made into acetyl-CoA. Acetyl-CoA inhibits the conversion of pyruvate to acetyl-CoA. Okay, so it actually inhibits glucose utilization by the brain. So again, ketone body production by the liver and its uptake by the brain prevents glucose utilization by the brain. It decreases it. And this spares glucose for the red blood cell, because the red blood cell can only utilize glucose because it doesn't have mitochondria. Okay, so that's what you need to know for starving condition. During acute starving or the first 24 hours, this is usually you're using the glycogen by glycogen phosphorylase through the action of glucagon. During prolonged fasting, gluconeogenesis kicks in, and there's three sources of gluconeogenesis: lactic acid, alanine, glycerol from the fat. The gluconeogenesis requires ATP. Where does it get the ATP from? From beta oxidation. However, these fatty acids cannot be utilized by the brain. Ketone bodies can during prolonged fasting, and this saves some of the blood glucose for the red blood cell. Okay. Now, this is showing you the fuels which you use during starvation. So acutely, or within the first few hours of starvation, you use all the blood glucose. Okay, so all of the blood glucose that you have in that you ingested during your meal, during your last meal, that gets used up. Then glycogen kicks in, this pathway. Then gluconeogenesis kicks in. Why is gluconeogenesis slow to occur? Because it's activated by gene expression. As I told you, glucagon and cortisol activate gluconeogenesis at the gene level. They activate an enzyme called PEP carboxykinase. And I told you that gene expression takes time. That's why gluconeogenesis takes time. Okay? Now, this is what your doctor said. Glucagon and epinephrine levels rise during an overnight fast acutely. These hormones, like insulin, exert their effects on the skeletal muscle, on the adipose tissue, and the liver. In the liver, they stimulate glycogen breakdown and the release of glucose. During a prolonged fast, hepatic gluconeogenesis is also stimulated by glucagon, but this response is slower than that of glycogen breakdown because it's activated by gene expression. The release of amino acids from skeletal muscle and fatty acids from adipose tissue are both stimulated by the decrease in insulin and an increase in epinephrine. Why? Because epinephrine activates hormone-sensitive lipase, which liberates fatty acids from triglycerides. Okay? These amino acids and fatty acids are taken up by the liver, where the amino acids provide the carbon skeleton. You remember I told you alanine is the source for gluconeogenesis, and the beta oxidation of fatty acids provides the ATP necessary for glycolysis. So alanine is the source of gluconeogenesis. The fatty acids are the fuel for gluconeogenesis. During a prolonged fast, levels of glucagon and epinephrine are even more elevated. Lipolysis is occurring rapidly, resulting in excess acetyl-CoA production. This excess acetyl-CoA is used to make ketone bodies, and so the levels of both lipids and ketones are increased in the blood during a prolonged fast. Muscle also use fatty acid as a major fuel, but the brain doesn't. Red blood cells have no mitochondria and continue to be dependent on glucose. More on the brain adapts to using ketone bodies for some of its energy, and this actually shuts off or decreases glucose utilization. Okay, this shift from glucose to ketones as the major fuel diminishes the amount of protein that must be degraded to support gluconeogenesis. Okay, so this requires some more explanation. Alanine is being broken down, and so muscles are being broken down to make glucose, essentially, and this gives rise to muscle wasting. If you see starvation countries where starvation rates are very high, you'll see people who are very, very thin and they have no muscle because this has happened. All of their muscle has been broken down to produce alanine, and alanine has been used to make glucose. And glucose, the main use of glucose is one of the main uses for glucose is to fuel the brain. So by utilizing ketone bodies and by decreasing its use of glucose, the brain is actually sparing some of the muscle wasting, because by decreasing its utilization of glucose, is decreasing the need for gluconeogenesis. By decreasing the need for gluconeogenesis, you decrease the need for muscle protein breakdown. So you decrease muscle wasting. Okay. There is no energy storage form of protein. Yeah. Okay. Any questions about the fasting state? No? Okay. Okay, so this is, okay, all good. So now this, you have to know this as well. These are the fuels which are utilized by your different organs when during well-fed conditions and during fasting conditions. The liver utilizes glucose and amino acids during well-fed states. It utilizes fatty acids during starvation conditions. It's important and essential for you to know that the liver does not utilize ketones because the liver lacks an enzyme called thiopherase. And thiopherase is required for you to utilize ketones for energy. The liver lacks this enzyme, so it doesn't use ketones. The liver makes ketones, but it doesn't use them, okay? Resting skeletal muscle uses glucose. During starvation conditions, it uses fatty acids and ketones. Cardiac muscle, the same thing. Adipose tissue, glucose and fatty acid, the same thing. These two are the ones you have to know as well. The brain during well-fed conditions utilizes glucose, also in fasting conditions, but during a prolonged fast, it begins to utilize ketones. Why? To spare glucose for the red blood cell and to decrease muscle wasting. Okay? The red blood cell, because it doesn't have mitochondria, can only utilize glucose. So from this lecture, you have to know some of the enzymes which insulin is activated, the mechanism by which it activates them. You need to know the distribution of GLUT proteins. You need to know that GLUT1 and GLUT3 are present in the brain and RBCs. They're insulin independent. GLUT2 is present in the liver, and it's insulin independent. GLUT4 is insulin dependent. It's present in adipocytes and skeletal muscles. You need to know that during a prolonged fast, the brain switches to ketone body utilization to spare glucose. Okay, you have to know that. So these are some questions which could come in your exam. Okay, thank you very much. Any more questions?